In this video, we are going to solve Pearson Edexcel International GCSE 9-1 with the paper code for Unit 4B1, the Science Double Award 4SD0, 1BR paper. Okay, it is a regional paper and it is the latest paper till now. It is available on 13 May 2025. It was released for the examination hall over there. Before starting the solution of this paper, I will say kindly subscribe my channel and share the video, like the video with your friends, and uh, comment if you have any confusion in any question over there. Okay, it is 110 marks question paper over there. If we go in detailed solution of this, I will say I will try to solve the paper without the use of any type of marking scheme or like that. If my answer is feeling you like it is only the conceptual paper solution as a teacher over there, and I'm not using any type of, I can say, marking scheme in front of me. So stay conceptual and I will try to explain in detail over there. Thank you. Let's start the solution. The photograph shows a fungus. Okay, it is a fungus called mucor growing on algal plate or agar plate over there. Cells within mucor contains a nuclei and mitochondria over there. Okay, which of these is correct? Which of these is correct statement about the fungus mucor? Mucor is eukaryotic organism with cell wall made up of chitin. Yeah, it's fine because prokaryotes is those which don't have the well-defined nucleus, but here he said in the above example that it contains the nuclei. So it is a eukaryote and well-defined nucleus is available and cell wall is made up of chitin because he has also given us the information that it is a fungus over there. In fungus, cell wall is made up of chitin, while in plant cells, it is made up of cellulose over there. And for this reason, I will say option A is correct for this. If I just check the other ones also, this is the answer like that. Is eukaryotic cell wall made up of cellulose? No, it is for plants. Is prokaryotic? No, prokaryotic? No for it. So remember that fungi are eukaryotes and their cell walls are made up of chitin, unlike plants which use cellulose over there. Let's move to the second part over there. Plant cells contain chloroplast, cytoplasm, and starch. Plant cells contain chloroplast, cytoplasm, and starch. How many of these are present in fungus, such as mucor over there? So I will say that how many of these are present in the fungus such as mucor? Cytoplasm is present. Starch and chloroplast are not present because they are saprophytic. They are not able to produce their own food. And fungal cells contain cytoplasm, but they don't contain chloroplast as they are not photosynthetic. And they store glycogen, not starch over there. So I will say only one is present over there. And we can say option B will be fine for it. Let's move to next part over there. The fungus in the diagram is a circular of radius 45 millimeter over there. Calculate the area in millimeter square of the circle by using your answer. Just placing the values over there, you are getting over there like area is equal to pi is having value 3.14 into 45 millimeter square. So it is, I'm taking square of it only, and 45 square is 3.14 multiply 2025, and you will get the answer as 63 point, just use the calculator over there, 6358.5, and for going to two significant figures, then I will say I'm just rounding it off for two significant figures by rounding off this 5. It is making this one as 4. So it is 6,400 millimeter square over there. This is the area which is being taken over there. Vigor can reproduce using asexual and or sexual reproduction. State two differences between asexual and sexual reproduction. So I will say here difference. Okay. Uh, difference between sexual and asexual reproduction. Okay. Asexual involves one parent. I'm writing here for asexual reproduction. It involves only one parent is involved for the production of asexual reproduction over there. And while sexual reproduction, sexual reproduction involves two parents over there. And one more difference which I can write that asexual reproduction is produces genetically identical offsprings, genetically identical offsprings over there, in which we can say they are just like clones over there. And here, while in sexual reproduction, they produce genetically very varied offsprings. Genetically varied offsprings over there. Springs over there. And I can also say asexual is only involve mitosis, it involves meiosis and mitosis also over there. We can give some other examples over there also being taken over there. Explain why it is an advantage for mucor to be able to reproduce using sexual reproduction over there. So I can write it about that sexual reproduction sexual reproduction produce genetically varied offsprings which are uh, which means that when variation comes over there, that if the environment changes, the environment changes over there, then we can say that some individuals may have alleles, that some individuals will have alleles which have alleles that make them resistant, over there towards any disease, and increasing the chance of and increases the chance of species to survive. Just take the screenshot so that you can read it and try to understand this concept that sexual reproduction genetically varied offspring produces, and which means that if the environment changes, then definitely some individuals will have the alleles that make them resistant, and this will increase the chance of the species to survive over there. Okay. Let's move to question number 2 now. The diagram shows the parts of human respiratory system with some structures labeled like lungs and structure X over there. And here, if I say what will be this structure, then X will be the structure splitting from the trachea into lungs over there. And that will be bronchus over there, bronchus like that. Let's see what he is saying over there. Which of these structures are labeled X? That alveolus, bronchioles, bronchus. It will be bronchus over there, where I am saying that the structure splitting from the trachea into the lungs is called bronchus over there. Okay. State why a lung is described as an organ. Because it is made up of several type of tissues, because the lungs, the lung is made up of several different tissues, several different tissues. And which type of tissues it can have? It can have epithelial tissues, it can have muscle tissues, it can have connective tissues like that, that work together, work together that work together to perform a specific function, to perform specific function. And which function is done by the lungs? Gas is called gaseous exchange over there. So the example which I have told you about tissues is related to epithelial tissues, it's related to muscle tissues, it's related to connective tissues also over there. Explain how contraction of the diaphragm causes the air to move into the lungs over there. Okay, for this one, I will add some step-by-step phenomenon. I will say first, and in the start, at the first step over there, the diaphragm muscle, diaphragm, the diaphragm muscle contracts, muscle contracts and flattens. After that, what will be the result of this? It will increase the volume of thorax, and thorax is a chest cavity basically. So use the scientific word. Don't use chest cavity, volume of chest cavity like that. Use the word, the volume of thorax over there. And in the result, what will happen? This increased volume causes, we can say, a decrease in pressure inside, causes a decrease in pressure inside the lungs as compared to the atmospheric pressure, to the ATM, atmospheric pressure, and outside over there. So when it is causing the decrease in pressure inside the lungs, then I will say air moves from an area, air moves from an area of high pressure, high pressure, like outside the lungs over there, to an area of low pressure, to an area of low pressure, which is inside the lungs over there, inside the chest cavity over there. So inhalation occurs. So inhalation occurs over there. So I have written the complete contraction of diaphragm causes air to move in. This is called inhalation, and inhalation is basically causing the taking in of oxygen gas for breathing purpose or for gaseous exchange over there. A scientist compared the rate at which oxygen is used during exercise by a person who is a non-smoker with a person who is smoker. Okay, the rate is measured in centimeter cube per minute for each kilogram of body mass. The table shows the scientist results. Non-smoker 35 is oxygen rate, while smoker is 27. So and the rate is given in centimeter cube per minute for each kilogram of mass of a body. So calculate the volume of oxygen used in 30 minutes by a non-smoker with mass of 70 kg. So I will say here that volume per minute, if I go for that, then it will be 35 multiplied 70. This will be for per minute because it is 35 and it is 70 kg. So it will be 0, 7, 5, 35, 3 carry 7, 3, 21, and 324, 50 cm cube per minute. And he is saying for 30, so I will just multiply 2450, 2450 multiply by 30. So it will be double zero, and 245 multiplied by 3 will equal to 735 cm cube total volume of gaseous exchange by using this rate for non-smoker over there. Explain the effect of cigarette smoking on the rate at which oxygen is used over there. So I will say cigarette smoke contains tar and other chemicals that damage the alveoli. Cigarette contains tar and other chemicals that damage the alveoli, alveoli, reducing the surface area of reducing the surface area for gas exchange, for gas exchange over there. And it also contains carbon monoxide over there, which binds irreversibly with to hemoglobin over there, hemoglobin over there, and in red blood cells, in RBC, red blood cells over there. And in red blood cells, so reducing the amount of, so reducing the amount of oxygen gas that can be carried, that can be carried by the hemoglobin over there. So this means the body cells will receive, so body cells will receive less oxygen, and we can say so the rate at which so the rate at which the rate at which oxygen can be used during exercise over there during exercise is lower in smokers over there, in a smoker. Okay, this is the reason which I told you that tar and other chemicals damage the surface of alveoli, and it is also having carbon monoxide, which is going to bind with the hemoglobin irreversibly over there, reducing the amount of oxygen to be present over there. Okay, let's move to next question over there. The diagram shows a human skin cell. Okay, ribosome, cell membrane, mitochondria, nucleus, and ribosomes are shown over there, in which of the labeled structures does protein synthesis occur? Remember that, just like a memory, that cell membrane is a selectively permeable, mitochondrion is powerhouse of the cell, nucleus is a CPU, like to control all the functions, and ribosomes are the organelles which are used for the production of protein synthesis over there. So this is the right option for this. Ribosomes are the sites of protein synthesis over there. After that, he is saying a cell has cells, all cells have cell membranes. Which of the labeled structures are also found in plant cells over there? So I will say here mitochondrion and nucleus only. Mitochondrion and ribosome only. Mitochondrion, nucleus, and ribosome. So mitochondrion, energy powerhouse, nucleus, and ribosome, everything is present in plant cell over there. Okay, let's move to the part B over there. The table shows the information about the number of mitochondria in different types of human cells. Skin, villus is the basic unit of stomach like that, a villi like that, and sperm is a male gamete cell over there. Number of mitochondria is in villus is maximum, and number of mitochondria in 10 micrometer cube of cell, and volume of cell in micrometer cube is 1700 and 2500 is given over there. Okay, what he is asking over there, what to do? He is asking the volume of villus. Volume of villus is 2500 micrometer cube. He is right over there. Calculate the ratio of the volume of villus cell to the volume of sperm cell, and answer in the form of number and one over there. So for that case, as he said, 2500 micrometer cube, and number of mitochondrion is for sperm cell is given in the form of 75 like that, and in total there are 12 mitochondrion in 10 cm cube over there. So I will say here that first of all, number of mitochondria per micrometer cube I need over there. So for that purpose, I will say here, I will just divide for 12 divided by 10 over there so that I can get for 1 micrometer cube over there. Then it will be 1.2 uh, mitochondria per micrometer cube in sperm cell will be available. So total volume of sperm cell will be total mitochondria over total mitochondria 75 divided by 1.2 over there. So I will say it will be approximately equal to 1 by 2, 12 is 75. So it is 62.5 micrometer cube. So this is the total volume of sperm cell over there. Total volume of sperm cell over there. Now I will have to go for ratio of volume of villus and volume of sperm cell over there. So I will say here that I should go with 2500 100 ratio 62.5. If I divide 1 ratio, then it will be approximately equal to 40 over there. So it is 40 ratio 1. Again, I am telling you here that I took this for 10 micrometer to 1 micrometer cube over there, and then I divided the mitochondrion with the this 1.2 number over there. I got 62.5, and then I took the ratio with 2500, which was the volume for villus given over there. Okay. Now he is saying using the information in the table, comment on the importance of mitochondria to activities of different cell types over there. So if we just look above over there, I'm just commenting, so I'm not writing over there. Just listen my words very carefully. Willis have the high, very high number of mitochondria, like 2000 over there, because they are involved in active transport of digested food molecules from intestine into blood. So active transport requires a lot of energy of ATP to produce by mitochondria over there. So this is having a very largest number over there. If I go for sperm cells over there, then I will say they have high density of mitochondria per micrometer cube, like 12 over there, as compared to villus and the other one. It is having the high density over there for the, so because they need a lot of energy for movement, swimming towards the egg cell like that. So the mitochondria are concentrated in the mid piece to power the tail over there. In case of skin cells, if you observe there, they have low number and density of mitochondria over there, low number of mitochondria and low number of density over there, because their metabolic activity is lower. Their main function are protection and barrier formation, which are less energy intensive over there. So by just observing this data, I have just commented. You can write it in your own words. And if you feel that you are not understandable, just take the video 2 minutes back and just listen again for this solution over there. Okay, let's move to question number 4 now. Here he is saying that fruit juice drinks contains a range of nutrients. The table shows some of the nutrients in the fruit juice drink and the percentage of recommended daily allowance RDA. Okay, is nutrient in 16-year-old. Okay, starch 2.8 400 cm cube of drink in gram. Okay, 400 for 2 for RDA. And sugar is 6.4 and it is 4 fat and fiber. Okay. Vitamins and minerals are not listed in the table. Name one other component of balanced diet which is not listed in table. So I will say that can be protein, or I can also add that can be water over there. Okay. Calculate the RDA of fiber for a 16-year-old. give your answer into the nearest one over there. So it is 9.2. It should be taken as 9.2 in mass of nutrient in 400 cm cube. It is 9.2, and percentage of RDA in this is taken as 35 over there. So I will say here 9.2 divided by 35 multiplied by 100 will equal to 26 gram over there. After that, he is saying state the function of the fiber in human diet over there. So I will say fiber adds, fibers means roughage as we eat over there, bulk to pieces, and which helps the muscles of intestine push the food along, push the food along, and preventing constipation. Preventing constipation over there. Describe how starch is digested into glucose in human alimentary canal over there. So digestion of starch begins in mouth. It begins in mouth, where we can say saliva containing salivary amylase, or we can say enzyme amylase breaks down the starch, breaks down the starch into disaccharides called maltose over there. And this maltose comes in small intestine, and in small intestine, pancreatic amylase continues this process, over there, where we can say the maltose is then broken down. The maltose is then broken down into monosaccharide glucose, and when it is broken down into glucose by enzyme maltase over there. And in case the maltase is present over there, which is produced in the which is produced in the lining of small intestine, in the lining of small intestine. So over there. So glucose is then absorbed into bloodstream. So glucose is then absorbed in blood stream over there. So it was the overall explanation in which glycogen, or we can say um starch, is digested into glucose in human alimentary canal, where it is assimilated into body over there. Okay, after that, he is moving towards DCPIP is a blue solution that turns colorless when enough vitamin C is added. Okay, a student uses the apparatus to compare the concentration of vitamin C in different fruit juices. Okay, 5 cm cube DCPIP solution, dropping pipette cleaning fruit juice. Okay, this is the student's method. Place 5 centimeter of blue DCPIP solution in a test tube. Fill it, dropping pipette with fruit juice. Okay, so fruit juice is falling, dropping pipette containing fruit juice over there. Adds drops of fruit to DCPIP. Record the number of drops of fruit juice added until DCPIP solution turns colorless over there. So he is saying the table shows apple 18 grab means the solution to colorless, how many drops were added from the fruit over there for apple 18, 22, lemon 4, lime 7, orange 3 over there. The graph shows the different concentrations of vitamin C on the number of drops needed to DCPIP solution to colorless over there. Okay, determine the concentration of vitamin C in lime juice over there. So from here, from the graph, if we observe concentration of vitamin C here, and we can easily say here that uh, from the graph, like 7 drops of lime juice correspond to vitamin C concentration like that. So I will say here 7 drops of lime juice from this given table like that are dropped into vitamin C concentration solution over there, 7 drops. So number of drops of fruit juice for lemon is 7 like that. So it is 5, 6, and 7 drops like that. It is being taken like this. And for this one, we can say concentration will be approximately equal to 30 and 34, 35, 34, 36, 38, and 40. So it is 36 milligram um, per cm cube over there. So it is 36 milligram per cm cube. 36 milligram per cm cube. Give the fruit juices in order of increasing the concentration of vitamin C. One has been done for you. So lime is at the top over there. So I will say the grape will be at top, grape, and then I will say apple. You can check the answer again over there. And then lime is done for me. And then I will go lemon and orange over there. So if you can observe there, it is given according to this arrangement over there. grape as top, apple down, then lime, then lemon, and at the end will be the orange over there. So from lowest to highest concentrations, lowest concentration is from grape to orange is going on. So fewer drops means a higher concentration of vitamin C, as less volume is needed to colorless or decolorize the DCPIP over there. So I think so this arrangement is understandable now for you. Explain how the student could modify the experiment to give more accurate measures of the concentration of vitamin C in fruit juice over there. So I will say use a pipette or burette. Use a pipette, PIPETTE, pipette or burette, RETTE, burette, and that can measure the volume more accurately. That can measure the volume more accurately than drops. So if you are counting the number of drops over there, it is not more precise over there. So I will say repeat the experiment several times like that. And repeat the experiment several times so that we can say uh, for each fruit juice and calculate the mean number of drops per volume several times and take mean of several drops of different volumes over there. So this is the way for 2 marks I have given the two reasons, like use the Purita pipette that can measure the volume more accurately and repeat the experiment for getting the volume of drops over there. It was your question number 4 over there. Okay, let's move to next question there. Okay, here it is now question number 5. Okay, where he is saying the diagram shows the human shows the diagram shows the human female reproductive system with some structures labeled over there. In which structure does ovulation occurs over there? So ovulation, if we remember towards this, we can simply say that ovulation is happening in the ovary where the release of an egg occurs over there. That process is called ovulation. So I will say that it is for X over there, which is an ovary over there. So I will say X in the form of option B should be gone with it, that ovary is shown with the help of X over there. After that, he is saying, explain the importance of progesterone in the menstrual cycles over there. So I will say progesterone is produced by the corpus luteum, the remain of the follicle after ovulation. Its main functions are, I will say, it maintains the thick blood rich lining, the thick blood rich lining over there, rich lining of the uterus over there, like endometrium, making it suitable for making it suitable for a fertilized egg, for making suitable for the implantation implantation of fertilized egg over there, and it inhibits it inhibits the release of the release of further FSH, SH, and LH hormones from the pituitary gland, from the pituitary pituitary gland, preventing further evolution during pregnancy over there. Preventing further evolution, during pregnancy over there. So this was the importance of progesterone in menstrual cycle over there. Explain how the placenta enables developing embryo to obtain nutrients over there. So I can say here it is for 3 marks. It means I will have to add at least 3 main steps over there. So placenta contains villi which are in close contact with the mother's blood in the uterine wall. Okay, I will say here the placenta has villi which contains wait a second, which villi which uh, are in close contact in close contact which are in close contact with the mother's blood, with the mother's blood in uterine wall, in uterine wall, uterine wall over there. Nutrients like glucose and oxygen diffuse from mother's blood. Nutrients and oxygen from mother's blood. Nutrients and oxygen from the mother's blood diffuse across placenta, diffuse across placenta, diffuse across placenta into fetal blood in the umbilical umbilical vein over there. And after that, I will add here a little bit that the the fetal and what can I say there? Maternal blood don't mix, but the close proximity allows for efficient exchange over there. All right, let's move to part number B over there, where he is saying for fertilization to occur, a sperm needs to reach in an ovum. Which of these describes the number of chromosomes in the nucleus of human sperm over there? So that will be haploid and 23 chromosomes, because the other haploid to make a diploid after fertilization is egg cell, which will also have the 23 chromosomes, because the total are 46. So option C will be much perfect for this, where I am saying haploid number of 23 chromosomes is available over there, because gametes are haploid. So remember that sperm and egg are the gametes which are haploid over there, and these haploids, when they mix by the process of meiosis, they make a diploid zygote, which can give the formation of new organism over there. After that, he is saying here that the scientists are developing a contraceptive drug to reduce fertility. The drug is tested on 25 male mice. The graph 1 shows the effect of different concentrations of the drug on over mean concentration of the sperm semen. Okay, this graph 1 and this is graph 2. Concentration of drug in arbitrary. Okay, it is showing approximate minimum 6 value. It is showing the minimum 0.5 value over there. Okay, what he is asking over there, the scientists conclude that the drug could be used to as a contraceptive to reduce fertility in humans. Discuss the scientist's conclusion. Use the information in the graph of your own knowledge to support your answer. Okay, I'm going to elaborate by statement, because again and again, scroll up, scroll down for writing purpose. I'm just ignoring the writing over there. Just listen very carefully what I am saying according to the given data representation over there. I will say here that graph 1 shows that the drug concentration increases, the sperm concentration decreases significantly from 8.5 million sperms, uh, from 8.5 million to 1 million, like that. And graph 2 shows that the drug concentration increases, and the percentage of motile is being taken from the sperm also decreases from 75% to 10%, like that. So both a low sperm count and poor sperm mortality are known causes of infertility in humans. So drug that causes these effects could work as a contraceptive over there. And if I go for limitations of this conclusion, I will say the drug was tested on mice, not humans, and human physiology may respond differently. And the sample size of 25 mice is quite small, and large sample size would be needed to be confident in the result over there. And the long-term side effects of the drug are unknown, and the graph shows the effect of at specific time points over there. Uh, so the long-term effectiveness is not known according to the given data over there. So this was a little bit explanation which I have tried to conclude according to the given data over there. You can also use them to make your own conclusion related to this. Let's move to question number 6 over there, where he is saying a student uses an apparatus to investigate the osmosis in non-living stem system. Okay, this student is method the partially permeable tubing at one end with a string. Okay, to make a bag. So fill the bag with sugar solution and the bag at the other end with string and measure the mass of the bag, string and sugar solution. Place the bag in test tube filled with the pure water. After 5 minutes, remove the bag and measure the mass and return the bag to the test tube. Repeat the measurement for 5 minutes for 30 minutes. Every 5 minutes for 30 minutes in total. So 0 to 30 minutes he is saying mass in of bag in gram, first of all, is going to increase from 15, 17, 20, 23, 26, 27, and at 30 minutes it is stopped over there. Now he is saying draw a line graph to show the student's result. Join your points with a straight line over there. So 0 to 530. Okay. 0, 10, no, 5, 10, 15, 20, 25, and 30 and 35 like that. And for y, he is going up to 27 again. So I will say here that it is 5, 10, 15, after that 20, after that 25 and 30 over there. Okay. Let's check what are the values at 0, 15. After that, he is saying after 15, it is 17, 20, 23. 17, 20, 23. At 5 minutes, it is 17, 15, 16, 17, 16, and 17. Then it is 20. Then it is 23, 21, 22, 21, 22, 23 over there. After 23, it is going towards 26 and 27. So it is 26, 26 and 27 like that. And again, it is going towards 27 there. So if I join like this, it is just like a curve with a positive gradient over there. And from here, the graph, I will say, remember that graph should be drawn with time or minutes on x-axis and mass. So it is time and it is mass. So I will say, and mass of the bag on y-axis, and the points should be plotted on the table and joined with a straight lines. And the line will curve upward steeply over there at first and then plateau after 20 to 25 minutes over there. Okay. So it is making almost a straight line, just like a plateau over there. Okay, what he is saying there, describe the change of in mass of bag during the 30 minutes. So the mass of bag is increased. Mass of bag is increased rapidly. Is increased rapidly from 0 to 20 minutes. From 0 to 20 mins over there, and the rate of increase and the rate of increase is slowed. Rate of increase is slowed between 20 to 25 minutes, between 20 to 25 mins over there, and the mass remained constant, and the mass remained constant, and the mass remained constant uh, from 25 to from 25 to 30 mins over there. All right, let's move to the next part over there. What he is saying, explain the change in mass of the bag during the 30 minutes over there. So bag contains overall a sugar solution and is placed in pure water, and water has higher potential and than the sugar solution over there. So I can say, as already the sugar is present in bag, so water from outside at higher potential is absorbed, and mass of bag is increasing, and when concentration becomes equal by osmosis, no more bag increase size of masses happening over there. And the student decides to investigate how increasing the temperature would affect the results over there. Describe how student can could repeat the experiment safely at higher temperatures over there. So I will say use a water bath, use a water bath for high temperature, and I can say wear safety goggles to protect the eyes from hot water. Wear the safety goggles to save eyes, or like we can say to save eyes from hot water. from hot water over there. After that, I can say use a thermometer to monitor the temperature. Use thermometer to measure and monitor the temperature over there. So this is the way how we should go for making our results more precise related to it. Explain how repeating the experiment at higher temperature would affect the results over there. So for this purpose, where he is telling me, I can say at higher temperature, water molecules have more kinetic energy and move faster. This would increase the rate of osmosis over there at high temperature. At high temperature, water molecules have high kinetic energy over there and move faster over there, and this would increase rate of osmosis. This would increase the rate of osmosis over there. And when rate of osmosis increased, so water would move into the bag. So water would move into bag more quickly, and so mass would increase faster rate. So mass would increase at faster rate over there. So when it will be increased at faster rate, so bed will reach its maximum mass in shorter period of time. So bag will reach at its maximum mass at very faster rate. So this was your overall explanation related that when at high temperature you are going on, what would affect the results? You will get them early over there. No more discussion about it. So here he is saying limpets are marine animals that attach to the rocks on the coastal shores. The diagram shows the food web that contains the limplets over there. Okay, limplets, algae, dog whelks, okay, herring gulls, starfish, and periwinkles, okay, which are the trophic level of the starfish in the food web over there. So I will say here that primary is algae, and you are asking for starfish. It is producer's primary level and it will be secondary level over there. So I will say it can be secondary or tertiary according to its food web over there, that they can be secondary or they can be the tertiary. Why I said tertiary over there? Look at here. This is producer, this is primary, and this is secondary. Fine. Or tertiary, why I said this? If this is producer, this is primary, this is secondary, and then it is reaching to starfish, then it can be the tertiary stage over there to be kept in mind over there. After that, he is saying what is meant by population. It's very easy that group of organisms organisms of the same species living in the same habitat living in the same habitat living in the same habitat at the same time. That is called population over there. Explain why only 1% of the energy in the algae is transferred to the starfish over there. So I will say here that energy is lost between trophic levels in several ways by preventing it from being transferred to the starfish like that. Not all the algae is eaten by primary consumers. He said for 3 marks. Okay, what I should write about it? I will say that all the algae is not eaten by primary consumers. Primary consumers is not eaten by all by the primary consumers over there, like limblets and periwinkles like that. So a large amount of energy is lost as heat. A large amount of energy is lost as heat in respiration over there in the primary consumers. In respiration by primary consumers over there. So for that purpose, we can say energy is lost in waste materials like feces and urines from the primary consumers. Energy is lost by the primary consumers in waste materials like feces and urine over there from the primary consumers, and some of the energy is used by primary consumers for movement and other life processes. The movement and other life processes over there. So energy is lost only you can get is 1% for secondary or tertiary starfish in this food web level over there. Okay, here he is saying some scientists think that the food web on rocky shores are the risk from an increase. So it scientists takes per centimeter square per meter square and the mean temperature of the same rocky shore. So the graph of scientists results is shown mean temperature is 12 to 18. Okay. And the number of limblets per meter cube is going to increase. And as the temperature is increasing, number of limblets is decreasing. Describe the scientists could determine the number of limblets on the rocky shore. So how can he do that? I will say here, place one or more quadrants randomly. Place one or more quadrants randomly and on the rocky shore on the a rocky shore and count the number of limplets over there and calculate the mean and calculate the mean and number of number of limplets per quadrant. And at the end, I will say here that scale up the mean, scale up the mean, scale up the mean over there to find the number, to find the number, to find the number per meter square and if the quadrant is 0.25 meter cube, then multiply the mean by 4 over there. Okay, let's move to second part. What he is saying there: give a factor the scientist should control when comparing the number of limplets per meter square each year. So he should check he can check size of quadrant, size of quadrant over there. He can go for the time of day the count is conducted, the time of day when he is going to count to be conducted over there. He can also go the exact location on the shore, exact location on the shore over there. Okay, now he is saying the scientists conclude that the combustion of fossil fuels is a risk to the food web. Discuss the scientist's conclusion, use the information in the graph and your knowledge to support your answer over there. Okay. Uh, I'm going to give the conclusion over there that the graph shows a clear negative correlation between the mean temperature and the number of limpets. So as the temperature increases, the limpet population decreases. So combustion of fossil fuels releases greenhouse gases, which causes the global warming and an increase in average temperatures. So if rising temperature causes limpet numbers to fall, this would reduce the food source for starfish and dog whales, so disrupting the entire food web over there. So for this one, the graph shows a correlation, but it does not prove that increased temperature is the cause for the decline in limpets. Other factors linked to fossil fuel combustion, like ocean acidification due to increased carbon dioxide, could be responsible for the decline, and other unrelated factors such as pollution or overpredation could also be causing the limpet numbers to fall over there. So I tried to explain in my words. If you want to note down this, then just give the pause into the video and note the answer so that you can easily understand. Also, if you have confusion during my statements over there. It was a complete explanation of this graph and the above data over there. Okay, this was for 5 marks, the question over there. Okay, this is the question number 8. He is saying golden rice is an example of genetically modified plant. Golden rice has been given a gene to make a substance called carotene. This gene was taken from daffodil plants. State the term for a genetically modified organism that has been given a gene from a different species that is called transgenic organism. Transgenic organism over there. Let's move to next part over there. Describe how named enzymes are used to transfer a gene from one species to another species over there. So for this purpose, I will say, okay, step by step, I'm going for trying to make in 3 to 4 steps over there. Like named enzymes used for transfer of gene from one species to another. How? Describe how. So I will have to go for description over there. I will say here restriction enzyme is available over there, is used to cut the desired desired gene out. Gene means the carotene gene, like that, out of the DNA of daffodil, of the DNA of daffodils. And then same restriction enzyme is used, and same restriction enzyme is used to cut the plasmid and plasmid of a small circular piece of DNA from the bacterium, like that. That is called plasmid over there, creating complementary stick ends over there. So I will say here that sticky ends, sorry, stick ends, then sticky ends, and the gene and plasmid are the gene and plasmid are mixed over there, and mixed together, and then the enzyme DNA ligase, the enzyme DNA ligase is used to join the gene into plasmid, the gene into plasmid, forming recombinant DNA, forming recombinant DNA over there. Carotene is converted into vitamin A after being consumed by humans. Non-genetically modified rice does not usually contain carotene. Vitamin A deficiency is common in some countries. Explain why growing golden rice may benefit the people in these countries. So I will say vitamin A is essential for good vision, a healthy immune system, and cell growth. So in countries where vitamin A deficiency is common, it can lead to blindness and increased susceptibility to disease. So, golden rice provides a source of vitamin A, which can help to prevent these health problems, especially in countries where rice is staple food over there. So, I will say the common diseases for this one is, which I told you, is uh, vitamin A, where he is saying vitamin A is responsible for to go for, if it's deficiency, if I talk about that, then it can go towards blindness, means vision control is being done with the help of vitamin A. So carotene or golden rice should be used over there. And there is going towards illness and health problems, and especially to the countries which are rich in staple food over rice over there. All right. So vitamin A is to control that. So we will go for golden rice to be used. So just why some people may not agree with growing genetically modified crops such as golden rice over there. So for that one, I will say that they consider about environmental concerns, environmental concerns can be added. If I talk about it, then I will say there is a risk that modified genes could spread to wild plant populations with unknown consequences for ecosystems. So environmental concern can be taken over there. I can also take over there that there is health concern, then I can say that some people worry that eating genetically modified foods could have long-term unforeseen health effects like allergic reactions over there. There can also be economic concern can be there. For that one, I will say that farmers may become dependent on large companies that own the patents for genetically modified seeds over there. So let's move to next part. What he is saying over there, chemical pesticides can be used in a rice field to kill the insect pests over there. Okay, insects, pests, carnivorous beetles, predators of insects, pests, lizards, high trophic level predators. Scientists count number of each type of organism in the rice field. The scientists then spray the field with pesticide, and the number of each type of organism in the field are then counted every 3 months for a year. The table shows the scientist result over there. Okay, like 490, 26, 493, okay. 25, 30, 175, and 280. Okay, carnivorous beetles are given. Okay. Comment on the effects of different types of organisms of spraying pesticides in the rice field. Okay, I will have to just go for commenting over there. Just listen my words very carefully for this one. I will say that there is immediate effect for 3 months, like the number of insect pests crashes dramatically from 490 to 25, showing the pesticide is initially very effective. And if I look towards the effect on predators like carnivores, like that, the number of carnivorous beetles and lizards also decrease because they are poisoned by eating contaminated prey. Bio accumulation, like that, and because their food source is reduced. And we can also say like recovery of pests after the initial crash, the insects pest population recovers quickly from 25 to 280, if you observe there. Okay, from 25 to 280 in 12 months. And because the predators that normally control them are now gone over there. So this creates a bottom and boom and bust cycle for pests, which is unsustainable and requires the repeated pesticides application over there. Okay, this was just like an explanation which I tried to do for you. Oh, there is also a second part over there. All right. State two reasons why using biological control is better for the environment than using a chemical pesticide over there. So I will say biological control is it is more specific to pests and does not harm other beneficial insects or the wider food web over there. Okay, so biological control is more specific over there, and we can say that the pests are less likely to likely develop less likely to develop any resistance to biological control, resistance to biological we can say control over there. So for this one, I will also say that biological control does not pollute the environment. Biological control does not pollute the environment. Meant over there. So it was the overall explanation related to this. Okay, let's move to question number 9 over there. Okay, here he is saying that the ability of human to taste a chemical called is PTC is controlled by a single gene. The ability to taste PTC is coded for by a dominant allele T. Okay, taste T, and not being able to taste PTC is coded by a recessive allele called T. State what is meant by the term recessive allele. So I will say a recessive allele is an allele that is only expressed then in in phenotype as physical representation over there if if two copies are if two copies are are present in the form of T like that for the taste over there. It is masked by the presence of dominant allele over there. Okay, the pedigree diagram, okay, shows the individuals in a family who can taste PTC and those cannot taste the PTC over there. So male who cannot taste PTC is this, male with who can with white, okay, and then female cannot and with white it can. Okay, so what are the genotypes of the individual 1 and 2 over there. So I will say here that he is going with being taken as not able to taste, so as a female. So it should go with C over there. Okay. For this one, I will say that individual 1 cannot taste PTC. If you observe in the black color over there, he cannot taste over there. And on the other hand, I can say if he cannot taste, so must be small t over there. On the other hand, he is saying individual 2 can taste, but they have a child individual 7. If you can observe their individual 7 who cannot taste PTC. So PTC, that should be small tt. So individual 2 must be heterozygous. So it should pass the allele over there. So it should be capital T and small t over there, which I have marked over here. Okay, he is saying determine the probability of the individual 3 and 4 having another child that can taste PTC and is male over there. So for this one, I will say here that the parents and show using the user genetic diagram also. Okay. I am using here that parental genotype which I have written for you is going with heterozygous, because if you observe the position 3 and 4 both are showing as a dominant taste over there. So I am saying the parents is T T and T T over there is being taken as a dominant because both are showing the taste over there. So parental genotype is written for you. So the gametes produced by both of them will be T, small t, and T and small t over there. So if I go for making a table like offspring genotype over there, then I will say T, T, and T, T. So I will say here that I should go for writing here that in this format, when T and T are matching, you are getting capital T, you are getting capital T, small t, you are getting capital T, small t, and you are getting both as a small t. So phenotype capital T show can taste, and TT can also these two. If I'm changing the color here, just observe this one, and this one can taste, and this small t is not able to taste over there. So probability of child that can taste will be 3 upon 4 over there. And if probability of a male child should be taken, so it should be 1 by 2 also over there. So just take 3 over 8 as a probability by using this genetic diagram over there. Okay, let's move to the next part, what he is saying over there. Okay, he is saying people who can taste PDC say that taste is bitter. Many poisonous plants also taste bitter. Explain why there are many people who can taste PDC in areas where there are many poisonous plants. Refer the natural selection for your answer. So I will say here in population there was a genetic variation in the ability of to taste PTC controlled by the T and T alleles over there. So I will say there is a reason behind that is genetic variation is available over there. And then I will say individual with the dominant T allele will could also taste the bitter poisons in the plants and would avoid eating them. And individual without T alleles like homozygous or recessive like that, if it is going with small t allele over there, then could not taste the poison and were more likely to eat the poisonous plants and die before reproducing over there. So therefore, the capital T, like taste allele, provide a selective advantage. Okay, capital T allele provides a selective, we can say variation or advantage, selective advantage, and was more likely to be passed on the next generation. Over many generations, this led to higher frequency for the taste of these areas over there. People who can taste PTC often find in the taste of vegetables very bitter, suggest why the recessive allele for not tasting PTC still exists in human population over there. So I can simply say about it, in modern societies, the ability to taste bitter is not such a strong selective advantage as food is safe and readily available over there, and the recessive allele may be maintained in the population through heterozygous carriers who have the advantage of being able to taste the poison but can also pass on the non-taste allele over there. So in areas without poisonous plant, there might be no disadvantage to being a non-taster, so there might even be an advantage and being able to eat wider variety of the nutritious bitter vegetables over there. Okay, let's move to next question over there. Oh, still there is a question. Okay, then okay, no problem. Fly larva will continuously move around the edge of a predatory dish when placed under a lamp. The diagram shows that fly larva is in a petri dish. Okay, design and investigation to find out if changing the color of the light from the lamp affect the speed of the larva move. Include the experimental details over there. Okay, this is an experiment question over there, in which I will say that a possible design which you can use for this one to investigate the effect of light color on the speed of larva movement. So I will add little bit here. Okay, I'm adding little bit. First of all, independent variables. I'm writing little bit for you. Independent variables. Independent variables, if we talk, so the color of light from the lamp, like red, blue, green, white, can be taken. Color of light. It will be just like, we can say it can be taken as color of light. That can be red, blue, green can be taken as independent variable. And for development dependent variable, I will say speed of larva. Speed of larva, which we can say we can take uh, in millimeter per second or centimeter per minute, like that can be taken. And the control variables for this one, control variables. This was independent and it was dependent variable. Now I'm talking about control variables over there. So for this one, I will say the species and age and size of larva and the temperature of the room and intensity and brightness of the light and absence of other light sources can be taken as control variables for this one over there. And apparatus which you will require for this one, you will require battery dish over there. You can be required with fly larva, and here can also be added with fly larva like lamp will be required, and we can say here different colored light filters like for colored LED bulbs over there can be taken. Different colored different colored light light filters over there can be added over there. And you can also take a ruler stopwatch and color marker, or like that marker pen marker pen over there, that can be apparatus for this one. So if I go towards a method, place a single larva in the center of petri dish and allow it to matize for 1 minute. Place the white lamp a fixed distance of 30 centimeter above the petri dish and use a marker to make a small dot on the edge of the petri dish to define the start point, and then start the stopwatch and the larva to cross the start point, and time how long it takes the larva to complete one full circuit of the petri dish. Calculate the speed using speed is equal to distance over time, like that. Repeat the steps for 1 to 7 as I told you over there, more and more to get the more accurate value, get the mean speed over there. Repeat the entire experiment like that. Handle the larva with care to avoid harming them, and compare the mean speeds calculated from each light color to determine which color results are the fastest and slowest movement over there can be shown over there. So this was your overall paper solution, which was a regional paper, and I tried to solve all the questions according to my knowledge. If you compare this solution with the marking scheme, there can be a little bit 1 to 10% variations over there, but it will be more precise and best solution of this paper. I think so you are going to understand it and trying to explain it on your own behalf. Thank you so much for watching this video. Kindly subscribe my channel. See you in the next video. Thank you so much.